Midterm grades were due on Monday, so they got turned in. Remember what I said, I wouldn't get real excited. We've only had about a third of the total points at this point that you'll end up having at the end of the semester. So that does tell you that the assignments will be a little more frequent and worth a few more points towards the end. If you just look in the grade book, that will give you your current average with everything, except when I double checked this morning, it looks like I didn't put up the grades for all the papers. So I'll have to add those later today. But those shouldn't change your average much because that was only 26 points. So considering we've already had more than 400 points, that might move it a little, but not a lot, most likely. Are there any questions about the most recent material we've covered on animals? Keep in mind, we're probably about 2 days away. Most likely by the end of tomorrow, we'll be done with animals, maybe Friday, depending on how quickly we move. And then I'll send you a set of questions with animals. This is really a long set because there are a lot of things that you're going to have to look up. So keep that in mind. Don't wait till the last moment to start on them. Are there any questions about what we've discussed with animals recently or anything else related to biology? Okay, if not, we're going to go to screen sharing, and we still have one more topic to look at with regard to animal diversity. And this has to do with humans and primates in general. If we look at primates and we look at human evolution, one of the things that we see about primates is that they have a couple of characteristics that distinguish them from other groups of mammals. Most primates are at least partly arboreal. Animals that are arboreal live in trees. Now, if you look at this chimpanzee, or if you've seen a chimpanzee before and you think about what the chimpanzee's face looks like, or if you think about a human face, notice that compared to a lot of mammals, chimpanzees and humans don't have a very big nose. And this has one big advantage for primates. This is a general characteristic that we see with primates. Since primates don't have a really big nose, your right eye and your left eye can both look at the same object. Now, there's a process that when we're talking about sensory organs, or even when we're talking about instruments like radar, called triangulation. And if you look at something with your right eye and your left eye, they're slightly separated on your face. So they're not looking at something from exactly the same point of view. Now, the advantage of looking at things from a right eye and a left eye that are separated is this gives you a better three-dimensional picture of the world, and it gives you better depth perception. And if you think about it, for animals like primates that live in trees, depth perception is crucial. You're trying to jump from one branch to another. It makes a big difference whether that next branch is 4 feet away or 6 feet away. You don't want to fall 40 feet to the ground if you think it's 4 feet away and it's really 6 feet away. Most mammals, think about if you have a dog or a cat at home, most mammals have a big nose that kind of sticks out in the front of their face. Well, one of the things this tells you is that for these other mammals, they rely much more on their sense of smell than they do on their sense of vision. But not only that, if you think about a dog, you know, the snout on a dog with the nose at the tip is so large. Dogs are mostly looking at separate things with their right eye and their left eye. They have a much wider field of vision than people do because their right eye is looking to the right, their left eye is looking to the right at the same time. Now, keep in mind, your brain has to be wired a little bit differently to interpret the visual information coming in. Because even though we talk about the eyes as seeing, it's really the brain that sees. Optical illusions don't trick your eyes, they trick your brain. So primates have what's called stereoscopic vision. Stereoscopic vision just refers to the fact that you look at the same thing with both eyes. But when we think about humans and how humans evolved from other primates, It's a good example of something we've been talking about all semester. The idea that we often look at biology with these preconceived notions about what's happening. And when we get trapped by these preconceived notions, it can lead us down wrong paths. Going way back before we had discovered really any fossils of primates and before evolution was accepted as an explanation in biology, and so there was this idea that humans had evolved from other primates, it was pretty obvious what separates humans from other animals and other primates. We're smarter than them. We have a bigger brain than them. This is what makes us different. Except it turns out that's not the biggest difference between primates and between humans, or between primates and mammals. So if we think about humans, how are some ways that humans are different from other animals, even other primates? Well, we've got a big brain. Other animals like chimpanzees do have a language, but our language is much more complex. Humans use tools much more than other animals. And again, chimpanzees do use tools. There are some other animals that use tools as well. Some chimpanzees wash their food before they eat it. They dip it in water to rinse it clean. So we have a big brain. We have language. We use tools. And another thing that is different is, if we look at most mammals, dogs, cats, deer, horses, most mammals walk on 4 legs, we walk on 2. Well, it's pretty obvious that what separates us from other mammals is the fact that our brain is so large. Except, as we've discovered fossils of primates that are now extinct, but human ancestors, when we look at the very first fossils that were discovered, their brain size is about the same as a chimpanzee's brain. And there's not much evidence that they used very complicated tools, any more complicated than a chimpanzee would use. Language is hard to tell, words don't really fossilize, but by looking at the bone structure, the earliest human ancestors walked on 2 legs, not on 4 legs. So when we look at the characteristics that we might think of as separating people from other primates, really the first characteristic to evolve was walking upright, or it's sometimes referred to as walking erect. The earliest human fossils that were discovered that walked erect were called Homo erectus. Today we've discovered earlier fossils than Homo erectus that walked upright. One of the first fossils to be discovered, one of the oldest fossils, I should say, not the first, but one of the oldest fossils to be discovered is a fossil that was named Lucy. And Lucy is a member of the genus Australopithecus. And Australopithecus means southern ape man, because the primates that were the ancestors of the line that eventually gave rise to humans tend to evolve in the area that today we would call South Africa, some of the areas there. Some of the later ancestors actually evolved more in the area that today we would call Kenya and Ethiopia. But southern ape man, Australopithecus, was so named because some of the early fossils were discovered in southern Africa. And if we look at Lucy, Lucy has a brain the size of a chimpanzee, but walks on 2 legs. By the way, everybody thinks, scientists are always very methodical and very organized and very non-emotional, but Lucy is a good example of how this isn't always the case. Does anybody know how Lucy got her name? Keep in mind, when Lucy was named, we didn't even know if she was a she. Well, they were doing an archaeological dig out in Africa. And when you do a dig like this, I did one semester at school, not in Africa, in Utah, but you basically poke around in the dirt and the sand until you discovered a fossil. And then you use basically a little, like a hand broom, and you clear off all the debris. And it's a very tiring, very methodical, and I hate to admit it, but very boring process, because most of the time you don't really find anything. And when the fossil was discovered that was named Lucy, what was playing on the radio? A song by the Beatles, Lucy in the Sky with Diamonds. So the fossil was named Lucy. Now, from Lucy and from other fossils, what we've concluded about human evolution is, out of those characteristics that separate humans from other primates, the first one to evolve was walking upright, walking on the hind two legs. Notice this picture shows a chimpanzee, and most primates do what's called knuckle walking. They walk on their front knuckles. They kind of make a fist, and they support most of their body weight on their front limbs, on what we would call the arms. Around the time that the earliest ancestors of humans evolved, the ancestral population seemed to be splitting into two groups. The climate in the part of Africa where they lived was drying up, and they had lived in the rainforest prior to that time. And in the eastern part of the range, as the climate dried up, trees need more rain than grasses. So the area that had been rainforest started to turn into a type of ecosystem that we call a savanna. And a savanna contains a lot of grasses, and every once in a while there are patches of trees in low areas where the water tends to collect, or along a stream bed. And so the ancestors of humans, we think, lived in the eastern part of this range. And as the climate became drier and trees lived further apart from each other, our ancestors had to kind of walk from one clump of trees to another. They still hung around trees quite a bit. Well, if you have to walk through grasses, and in the savanna, the grasses can be tall, 2 or 3 feet tall. Nobody's mowing the lawn regularly. If we look at the savanna, it's an advantage to walk on your hind legs, because if you walk on your hind legs, you're taller. And this means that you can see predators that might be hiding in the tall grasses. If you're down low and you're underneath the level of the grasses, you can't really see very far in front of you. So our ancestors started walking on 2 legs, probably for protection, as they went from one clump of trees to another clump of trees. But it turned out that walking on your hind legs had an advantage. If you're like this chimpanzee and you knuckle walk most of the time, your hands are busy. You're walking on them. If you walk on just the back 2 legs, your hands are free. And when your hands are free, when you're not walking on them, you can use tools much more easily. And if you use tools a lot, it's very helpful in an evolutionary sense. You'll survive better, you'll reproduce better, if you can tell other individuals in your population how to use tools. So using tools led to languages. And language requires a big brain. So the big brain evolved because of language, which evolved because of tools, which evolved because of standing up instead of walking on all fours. So in a way, the early ideas that we had about human evolution are really kind of backwards to what we have evidence occurred now. Something as simple as walking on 2 legs probably led to a chain reaction where walking on 2 legs increased tool use. This increased the complexity of the language. This forced evolution of the brain. So, good example of why we have to do experiments, and why when we look at, some of the early ideas that civilizations in classical Europe, the Romans and the Greeks, some of the ideas they had about nature turned out to be big misconceptions. And one of the reasons that they were misconceptions was because they didn't really do experiments. They didn't really look at evidence. They believed that you could answer scientific questions just by thinking hard about them. And today, one of the things we know is that if you just think about scientific questions and you think about answering them logically, the big problem that we encounter is we have all these preconceptions that cloud our judgment and lead us to the wrong conclusions. So when we think about human evolution, the big brain is something that's important in terms of making us different from other mammals and even other primates, but the big brain was not what separated us from the other primates. It was probably just walking on two legs. Well, throughout our discussion of animals, we've been talking about evolution. And we've been talking about how evolution is also connected to development. So what we want to look at now is animal development. So you should be looking at a slide with several different stages of animal development, starting with an unfertilized egg, going to something called the late gastrula. So let me know if you're not seeing that. Notice when we look at animal development, all animals go through the same stages. And at least up to the point of the early gastrula, if we were looking at gastrula, or this stage called a blastula, or a 4-cell stage, if we were looking at this in a human embryo and we were comparing it to, say, a fish embryo, or we were comparing it to a grasshopper embryo, or we were comparing it to an earthworm embryo, they would basically all look the same. Once we get to the late gastrula and beyond that point, then the body plan starts to diverge. Mammals start to look different than reptiles. Chordates start to look different than insects. So notice if we look at our unfertilized egg, we can see a couple of things. Now keep in mind, this is an egg cell, so the entire structure is one cell. We can't really make out any detail in the cytoplasm, but you should be able to see kind of a thin line here, a circle. Notice that this is the nuclear envelope. And inside the nuclear envelope, this little circle is the nucleolus. Now, the structure becomes more opaque. Light doesn't go through it as well. So when we look at it in a microscope, once the egg has been fertilized and forms what we call a zygote, it typically looks a little bit darker. Notice that there is kind of a little bluish green fringe around the unfertilized egg. And here we can see it a little bit better, but there is a structure that we call a membrane that holds all the cells in an embryo together. Now, once the egg is fertilized, once we have a zygote, this zygote starts to divide by mitosis. So remember, in mitosis, the cell replicates its DNA, then cytokinesis occurs, and basically, if everything goes right when the cell replicates its DNA and then divides by mitosis, these two cells will be genetically identical. Now these cells divide by mitosis, and then they divide by mitosis again. Now, once we get to about 16 cells, these cells start to, remember, we've got this membrane here. This membrane traps some fluid in the center. So what we actually see is a hollow ball of cells. And this hollow ball of cells is called the blastula. You might remember when we first started talking about animals, one of the things we mentioned was that all animals develop from a stage called the blastula. Now notice that the cells around the outside look much darker than the cells around the inside. If we would look at this blastula from a three-dimensional perspective, this blastula actually looks kind of like a beach ball. So think about a beach ball where the inside is actually hollow. Remember, we said that this ball is filled with a fluid, but it's hollow in terms of no cells. The reason that the edge looks darker is if you look right on the edge of the beach ball, you're actually looking through several layers of cells, right as you look around that curvature of the beach ball. If you look in the middle, you're only looking through 2 layers of cells, the one that's on the side facing you and the one that's on the side on the very back. So the cells around the edges look darker because when you're looking at things through a microscope, things that look lighter are things that more light passes through. Things that look darker are things that absorb more light. So the edges look darker simply because there's more layers of cells, they absorb more light. Now, if we look at the blastula, at some point, a layer of cells starts to push into the middle of the blastula. And when these cells start to push into the middle, this is the beginning of a process called gastrulation. And gastrulation is the process that forms your basic body plan. When we look at this, there's an opening. Keep in mind, we're looking at something three-dimensional. So when you look at this gastrula, this is really an object that's shaped kind of like a vase. So the opening to the vase is down here. And this opening, remember, it started out as a blastula. This is called the blastopore. And this opening is going to be either the mouth or the anus of the digestive system. So in animals that are protostomes, this is going to be the mouth. In animals that are deuterostomes, this is going to be the anus. So protostomes, another opening is going to develop, that'll be the anus. Deuterostomes, another opening is going to develop, that'll be the mouth. So this layer of cells that's on the outside of our vase, because remember, this is a hollow fluid-filled structure. So there's a layer of cells on the outside. This is called the ectoderm. The layer of cells on the inside, this is referred to as the endoderm. Now, in some animals, not all animals, it doesn't happen in sponges, it doesn't happen in jellyfish. After a period of time, a third layer of tissue starts to pinch off. Sometimes it pinches off the top of the endoderm, as is shown here. These are all slides of a starfish embryo. Sometimes it actually pinches off down here at the bottom, right by the blastopore. But this third layer is going to form something called the mesoderm. Meso means middle. So the ectoderm is on the outside, the endoderm is on the inside, and for some animals, there's a mesoderm in the middle. And these three layers, the endoderm, ectoderm, and mesoderm, are called embryonic germ layers. And these ultimately are going to produce all the tissues in your adult body. So if we look at mammals, as well as other chordates, this is kind of the pattern that we see. So let's look at the ectoderm first. The ectoderm ends up making something that we call the epidermis of your skin. The epidermis is the part of someone's skin that you can actually see when you look at them. There's a layer underneath the epidermis called the dermis. What does epi mean? Does anybody know in biology? There's epidermis. Epi means above. So the epidermis is above the dermis. It also makes the epithelial lining of the mouth and the anus. So the very beginning and the very end of the digestive system. It also makes up the cornea and the lens of your eyes. Now notice, these in a sense are all kind of logical. They're on the outside of the body. But what might not be as obvious is that the ectoderm also makes up the nervous system of the body. And it also makes up the adrenal medulla. You might have heard of adrenaline. Adrenaline is made in something called the adrenal glands. And the medulla is the middle part of the adrenal glands. Makes up the enamel of your teeth. And it makes up the epithelium of two glands that are associated with the brain. One is the pineal gland and one is the pituitary gland. The pineal gland makes something called melatonin that you might have heard of. It's a hormone that helps to regulate the wake-sleep cycle. And there are a lot of sleep aids that contain melatonin in them. The pituitary gland is sometimes called the master gland, and it makes a lot of hormones that control other parts of the body, especially other endocrine glands. So if we look at this, there are several parts of the body that are made by the ectoderm. Now, even though the mesoderm is the last embryonic germ tissue to develop, the mesoderm makes up the majority of your body. If we would look at the parts of your body by weight that come from the ectoderm and the mesoderm and the endoderm, most of your body weight comes from the mesoderm. So this makes up your notochord. Remember, we're chordates. Now, in us, in the vertebrates, the notochord disappears after a short period of time in the embryo, but the notochord is replaced by part of the skeletal system, the vertebrae. So your skeletal system comes from the mesoderm. Your muscular system, the muscular layers of your digestive system. This says your excretory system. We would call this your urinary system in most modern terminology. Your circulatory system, the blood vessels, the heart. Your lymphatic system. Okay, the lymphatic system is a weird system. Does anybody know what the lymphatic system does? You've probably heard of lymph nodes, but does anybody know what the lymphatic system actually does? Or
Yes.
Go ahead.
Oh my God. Oh, sorry.
Oh,
I
no
didn't know
problem.
I was
No problem. I thought you were answering the question. I wanted to
Oh
give you a
no, someone
moment
was
to think.
speaking.
No, that's good. Don't worry.
Do they help when, like, I think they're part of your immune system and they like fight off colds or something?
Yes, the lymph nodes do that, and the lymphatic system really has two parts. You may not be aware, but about 2 quarts of fluid actually leak out of your blood every day. And the lymphatic system has some vessels that pick up this fluid and then return it to the blood. And then you're right, while it's returning it to the blood, all this fluid goes through your lymph nodes and it basically fights infections there. And lately you've heard a lot of talk about, vaccinations, and if you get COVID-19, are you immune afterwards? Your immune system, after you get any type of infection from a bacteria or a virus in this case, but even a parasitic worm, your immune system makes some things called antibodies. And most of the cells that make antibodies sit in your lymph nodes. And basically, as the blood goes through, they check your blood and they make sure that there's none of the foreign, whatever, bacteria or virus or parasitic worms. They make sure that there's nothing in there that there shouldn't be. So you're right, this is an important part of the immune system. It makes the reproductive system, except for the germ cells. Now keep in mind, germ cells is another name for gametes, or eggs or sperm, however we'd like to phrase it. The mesoderm makes up the dermis of the skin. So the epidermis is the very outer layer. The epidermis is about 20 cell layers thick. The dermis is underneath that. It makes up the lining of your body cavity. The lining are some things called the pleura that go around the lungs, and the pericardium that goes around the heart, and the peritoneum that goes around your abdomen. And it makes up the adrenal cortex, the very outer part of those adrenal glands that we were talking about before. So notice, for some of the organs of the body, when you look at them, like the skin or the adrenal glands, different parts come from different tissues in the embryo. And then we have the endoderm. And notice that one of the best ways to think about the endoderm is, says the epithelial lining of the digestive tract, the epithelial lining of the respiratory system, the lining of this is all the urinary system and the reproductive system here. Basically, notice these are all the systems in your body that open to the outside: the digestive system, the respiratory system, the urinary system, the reproductive system. So you could think of all these systems as being kind of like a hollow tube that goes into your body. So the inner lining of all these hollow tubes, that comes from the endoderm. And then there are a few glands that come from the endoderm as well: the liver, the pancreas, the thymus, the thyroid gland, and the parathyroid glands. And notice this isn't insignificant, because when we look at the liver is often considered to be the largest gland in the body. Sometimes it's considered to be the largest organ in the body, although some people say the largest organ is really the skin. Now, how do we get from 3 basic tissues to all these different types of tissue? An adult human has about 240 different types of cells in your body. And if we would look at all the cells in a blastula, all the cells are really equivalent to one another, in the sense that, we just have a single layer of cells here. One side of each cell is facing the outside environment. One side is facing the inner fluid-filled part of the blastula. But even at this point, all these cells are not the same. If we would look at chemicals in the cytoplasm, they're not identical at this point. So what we want to see is look at what happens as development proceeds. And in embryonic development, there's 3 basic processes. There's cell division, and we mentioned this briefly before. Cell division happens by mitosis and cytokinesis. So if you look at your body right now, I mentioned you have about 240 different types of cells. If mitosis, if replicating your DNA and then dividing your DNA up went correctly, every cell in your body has exactly the same DNA. It has exactly the same 46 chromosomes, 23 that came from your mother in the egg cell, 23 that came from your father in the sperm cell. So no differences in any of your cells in the genetics. Now, there is one exception to this, which is if you're a female, in your ovaries, egg cells don't all have the same DNA. If you're a male, sperm cells don't all have the same DNA. And that's why when you have children, if you have multiple children, unless you happen to have identical twins, they don't look exactly alike. Because, if I have a child, it's 50-50. I got one version of chromosome number 1 from my mother. I got one version of chromosome number 1 from my father. And they don't have exactly the same alleles on them. So depending on which one I pass down to my child, one child might not get the same chromosome as another child does. Why do identical twins get the same chromosomes? How are identical twins produced? Does anyone know?
Because it splits.
Okay, what splits?
The zygote.
Okay. So if we look at an identical twin, we have one unfertilized egg. It's fertilized by one sperm. And somewhere in most mammals, this has to happen before we get past the 8-cell stage. So maybe 3 of these cells break off and form a separate clump, and the other 5 cells break off and form a second clump. Those can both develop into a complete embryo. So that's how we get identical twins: one egg fertilized by one sperm. Now, once we get to 16 cells, for some reasons we'll talk about in a few moments, if the cells get torn apart, they don't develop into a complete embryo, because they actually, by the time that we get to the 16-cell stage, we can't really see it very clearly, but cells have already started to specialize a little bit. Okay, so that's how we get identical twins. How do we get fraternal twins? Does anybody know how we get fraternal twins? And notice you could have identical triplets, or if you think about it, if all 8 of these cells broke apart, you could have identical octuplets. Not very likely, but possible. For fraternal twins, in fraternal twins, when a woman ovulates during her menstrual cycle, she releases 2 eggs, or she could release more than 2. And so the 2 eggs have different genetics because remember what I said earlier, you know, a woman could put in the chromosome number 1 she got from her mother, the chromosome number 1 she got from her father, and the same thing for chromosome 2, chromosome 3, chromosome 4, all the way down to her X chromosomes. So her 2 egg cells that are released during ovulation will have different genetics, and they're fertilized by 2 different sperm. So really, genetically, they're no more similar than any other pair of siblings are. Now, they're usually more similar for a different reason. Even though their genetics is really the same as an ordinary pair of siblings, they're in the mother's body at the same time, and they're raised in the family at the same time. So family circumstances change over the years. I have one brother who was born when I was 15, and my other brothers always complain, man, he got to eat at McDonald's all the time. We never got to eat at McDonald's or anything. So, you know, family circumstances change, and the environment in the mother's uterus is the same. So fraternal twins are usually a little more similar than ordinary siblings. Now notice, if you're a twin or if you have twins in your family, ordinarily about 1 in every 80 births or so is some form of twin. There are several other forms of twins besides identical twins and fraternal twins. They're much rarer. But if you're a twin, or if you have siblings or twins, now your odds of having twins are much higher than in a regular case. So it seems to be inherited. A few years ago, I had a woman in class, and she was one of 3 pairs of twins that her mother had, and she had 2 sets of twins herself. What is the accepted world record for the most number of children that a woman has ever had? Does anyone know? There is one woman in Russia who is, I guess I'll say credited is the right word, who is credited with having 69 children. As you can guess, she had twins and triplets mostly. That's not 69 single births. There must be something in the sperm that influences multiple births as well, because this woman eventually died. Gee, that's surprising, right? I think having 69 children would kill most women. But after she died, her husband remarried, and his next wife had, trying to remember, I want to say 19 children. So there must be something in the sperm that leads to multiple births as well. So what happens during embryonic?
Survive past childhood?
What?
You know how many children survive past childhood?
I'm not sure, because this was about a century ago in Russia. And if you go back to that time, what we call the child mortality rate, how many children die before adulthood, was typically around 40%. So I'll have to check and see if I can find that for you. I don't remember having read that.
Okay, thanks.
So we've got ordinary cell division. I'm sorry, do you have something else, Nick?
No.
Okay. So we have cell division by mitosis and cytokinesis. And we have differentiation, which is how cells become specialized. Now notice differentiation is going to be a genetic process. And then we have morphogenesis. Morpho, whenever we see that in biology, morpho refers to anatomy. So in morphogenesis, the cells and then the overall body takes on its shape, as well as the individual organs within the body. So let's look at some of the factors that affect this. So let's look at the structure of a sperm cell. So here's a sperm cell, and we have 3 basic parts. We have the head, the midpiece, and the tail. And the tail is a flagellum. The midpiece is a structure that mostly contains mitochondria. And the midpiece mitochondria are going to carry out aerobic cell respiration and provide the energy to wiggle the flagellum so that the sperm can swim to the egg. The centriole up here is what the flagellum is attached to. But if we look at the head of the sperm, notice that this whole bluish gray area in here is the nucleus. There's really no endoplasmic reticulum, no Golgi apparatus, none of that stuff that we typically find in the cytoplasm. There's a nucleus. If we're talking about people, there are 23 chromosomes in this nucleus. One copy of chromosome number 1, one copy of number 2, down to one copy of number 22, and one copy of the sex chromosomes. Around the nucleus, there's this structure called an acrosome. And the acrosome has enzymes, so that when the sperm cell encounters the egg, this acrosome bursts open and these enzymes are released and basically dissolve the protective layers around the egg so that the sperm can get in and fertilize the egg. However, if we look at eggs, it's a different story. Eggs have more cytoplasm than normal cells. So they have all the normal organelles. They have endoplasmic reticulum, they have Golgi apparatus, they have everything that cells need. They have ribosomes. They also have a lot of pre-made proteins. Now notice, since the egg is produced in the mother's body, these pre-made proteins are proteins that were made according to the mother's genetic instructions. There's also messenger RNA that can be used to make other proteins immediately. And again, notice that this messenger RNA comes from the mother. So this is really messenger RNA that was built according to her genetic instructions. And there are some other substances as well that can activate portions of the DNA or deactivate portions of the DNA. And all these things are called cytoplasmic determinants. They're out in the cytoplasm, and they're going to determine what type of cell the cell that they end up in becomes. And one of the keys is that these cytoplasmic determinants are not evenly distributed. So let's look at an example of this. In this diagram of a egg cell with cytoplasmic determinants, it shows two types of determinants. Notice in real life, there's a lot more. This is just for simplicity. So we've got some round kind of orange determinants, which it's showing mostly on the left-hand side of this egg cell. And we have some green triangular determinants, and it's showing them mostly on the right side of the cell. So once the egg is fertilized and mitosis begins, notice that we end up with some cells that have mostly green cytoplasmic determinants on the right. Notice there is a few of the orange determinants in here. And on the left, we have a cell that has mostly the orange cytoplasmic determinants. There are a few green ones in there. So this is a basic illustration about what happens. It's much more complex. There are dozens, if not hundreds, of these cytoplasmic determinants. So it's a little more complicated, and they're not divided quite as one-sided as this makes it appear. But depending on what sort of cytoplasmic determinants a cell has, and notice we're talking about when it gets to that 8-cell stage, the reason these are called determinants is they're going to determine the cell's fate. Think of it like career counseling. These determinants are going to tell the cell what it's going to grow up to be, what type of cell it will be, whether it's going to be a muscle cell, a skin cell, a brain cell. And the key thing is that these determinants work by regulating gene expression. Now remember from Biology 105, gene expression refers to whether a gene is actually being used to make proteins or not. So basically these determinants can turn genes on so that the cell starts to use them to make proteins, or they can turn genes off so that the cells don't use them. Now, if you think about what generally happens during development, most of these determinants, or I should say many of them, I don't know if I want to say most, but many of them turn genes off. And this is why. Think about a human. Well, humans have 46 chromosomes, or we can say 23 pairs if we would prefer. And if we look at those 23 chromosomes, there are somewhere between about 22,000 and 23,000 genes. Now remember that a gene is the instructions that tells a cell how to make a protein. So there's about 1,000 genes on a chromosome, on average. So people have approximately 23,000 chromosomes in a cell. But if I would look at one of your cells, if I would take a, well, somebody mentioned before that the lymphatic system plays a role in immunity. So there are cells called lymphocytes. If I would look at a lymphocyte, or if I would look at an simple squamous epithelium cell, which is the types of cells that we find in your epidermis. Or if I would look at a neuron, the type of cell that we find in your brain. Or if I would look at a hepatocyte, the type of cell that we find in your liver. Any cell I look at in your body, I would see that that cell uses approximately 10,000 genes to make proteins. Well, wait a minute, I just said that on the chromosomes, there are 23,000 genes, but a typical cell only uses 10,000 genes. So what happens during development is about 13,000 genes get turned off. The genes that are left that still work, that's what makes a cell into a certain type of cell in your body. So let's think about one protein that everybody might have heard of. There's a protein called hemoglobin that's found in red blood cells. Does anybody know what hemoglobin does in red blood cells?
It transports oxygen.
Right. It transports oxygen. It picks it up in your lungs, blood, goes all around the body and drops off oxygen. Now, every cell in your body has the gene for hemoglobin, but it's only expressed, it's only used to make proteins in red blood cells, in lymphocytes, in hepatocytes, in neurons, in simple squamous epithelium, in, the other 200 odd types of cells in your body. That gene is deactivated. It's turned off so that it's not used to make proteins. So what makes these 240 different types of cells in your body different from each other? Which 10,000 genes they use to make proteins. That's what makes one cell different from another. So during development, cells gradually, our genes gradually get deactivated. And then the genes that remain, the genes that that cell uses to make proteins, that's what turns it into a certain type of cell, either a red blood cell or a hepatocyte or a simple squamous epithelium or a neuron, whatever we're talking about. If we would look at the actual genes being used to make proteins in any of those, they're a little bit different. And that's what makes them the type of cell that they are. And if we look at all eukaryotic organisms, in eukaryotic organisms, what we see is, in general, there are more genes there than a cell uses. Now, keep in mind, bacteria are different. In bacteria, pretty much every gene in a bacteria cell is used to make proteins. But if you think about what I just said, a typical eukaryotic cell makes about 10,000 proteins, give or take. Some make a few more, some make a few less. If we look at bacteria cells have about 10,000 genes, but they use them all to make proteins. Now, the very simplest bacteria cells, well, at least simple in a genetic sense, only have about 4,200 genes. There are no living organisms that we know of that have fewer genes than that. So that appears to be kind of the minimum recipe, if you will, for having something that's alive. There is a lab that's taking these simplest bacteria and creating what are called knockout genes. We have the genetic technology now. We can go in and we can deactivate genes. And what this lab is doing is taking this bacteria that only has about 4,200 genes, and they're gradually deactivating genes by one. They're trying to see how simple an organism could they have that would survive. Notice a lot of times when they knock out a gene, the organism doesn't live, the bacteria doesn't survive. So maybe you can get by with a few less genes than 4,200, but at least right now that seems to be about the minimum. And if you don't use 4,200, then 10,000 seems to be the round number that we can use for most cells. So most cells use 10,000 genes. If you have a multicellular organism like the human body, which 10,000 genes you use, that's what determines what type of cell you are. So any questions about this for now? Okay. I will let you go, and I will see you again tomorrow, and we'll finish up talking about animal development, and we still have one other slight topic to talk about after that. We'll see if we can finish up animals completely tomorrow, but if not, we'll finish up on Friday. So if you have any questions, feel free to ask them. Otherwise, you can go ahead and leave, and I'll talk to you tomorrow.
I see you tomorrow.
Bye-bye. See you tomorrow.
Bye, sir.
Goodbye.
Thank you.
You're welcome. See you tomorrow.
Oh, Professor, I was able to make my appointment yesterday for my
Oh,
vaccine.
congratulations.
Yeah.
Not
We'll
too long to
see.
wait, I hope.
Yeah, in April. So not too bad.
No. Well, good. Then
Yeah.
hopefully you can get back to work and start making a little bit of money.
Yeah. It'd be great.
Good.
Well, you have a good day.
You too.